Good afternoon. Welcome to the second series XenoTech Drug Metabolism Seminar Series. This year we are focusing on the importance of ADME and DILI in drug development. My name is Tristan Thomas and I'm your local rep. And I actually cover the territory from here all the way down to Florida. Second series XenoTech concentrates on DDI and drug metabolism studies, and that focus, along with over 25 years of experience, provides you with the expertise needed to move your drug candidate through the pipeline and avoid potential pitfalls that can arise if you do not understand the impact of interactions and metabolism before you get into trials with human volunteers. It is for this reason that we consider it our goal to educate and inform about this important part of safety testing. Our first speaker is Dr. Griff Humphreys. He's our current principal at Aranmore Pharma Consulting, and he's spent more than 25 years in pharma at BMS, leading the Biotransformation Department. He is an expert in many aspects of metabolism, has authored over 100 publications, and serves on the society boards, including ISSX. Please welcome Griff Humphreys.
Good afternoon, everyone. We're going to do this without microphones, so hopefully everyone can hear me well enough. I think I can probably project fine. Thanks for the introduction. Thanks for coming out this afternoon. Kind of in full disclosure, I'm an independent consultant, but I'm on the scientific advisory board at XenoTech. This is a sponsored symposium, but what I'll present today really is my thoughts on this overall topic, and I will give you a balanced view of what I've kind of gained out of many years of experience in the field, both from a big pharma side and a consulting side. So hopefully this will be a useful talk for people. It's not kind of the typical presentation that I give, or I don't think that people hear very often, where you kind of would delve into one specific scientific area and cover it more in depth. What I'm trying to do in this presentation is give a broad overview of what this important section of a regulatory document consists of, what the studies are that are expected there, how you actually develop the section, the section 2.6.4 over time, and the progression of that section from really as you discover compounds all the way through to where you might register a compound. So what I'll talk about today is really the generation of ADME data for a new chemical entity throughout development phase. The contents of a final filing package, that's kind of what we'll first start with and then work backwards. Considerations of what ADME data should be contained at the time of the IND. So that's where I think it's a little less clear and probably where people, especially from smaller companies, have a lot of questions about how to build that IND package for the 2.6.4 section. So the types of ADME data that are typically found in INDs, interdependencies with other sections within the non-clinical and clinical sections of those first regulatory documents. And then I'll give some additional detail on human dose projections and DILI liability projections. So here's the full picture of exploratory discovery all the way through to commercialization and launch for a drug. And some key milestones depicted here on the top, the IND filing, end of phase 2 meeting, and the NDA filing. And this is just a couple of key packages or times throughout the process where different types of ADME data are generated. It starts with the early discovery screening, where candidate optimization really begins. You start oftentimes looking for liabilities, doing early PK assessments, seeing what kind of challenges you have in the optimization phase and how you might be getting over those challenges. And the goal really being optimization of PK and the minimization of liabilities. So as you progress out of discovery into candidate selection and start to perform IND enabling studies, you get into this really candidate characterization phase. And this is where you've got a single compound, perhaps one or two that you're choosing from, and you're really starting to get into detailed characterization. And at the end of this, you'll have enough data to file an IND. After that, then you move into a section which would be dominated by the clinical studies and the non-clinical studies associated with those, and really getting into full characterization of a drug. Finally, and this is a key endpoint really for the 2.6.4 section, is the end of phase 2, where you want to have pretty much the entire package in place, and then you can really successfully move into the large clinical trials. But sometimes there's still gaps at that point, and they have to be filled for the final NDA package. So what I'm going to start on is just giving you a flavor for all the data that goes into this kind of final package, the end of phase 2 or the NDA section 2.6.4. So I'll go through actually the various sections. If you don't know, the 2.6.4 is broken into the different components of ADME. But there are some extra components. One of them is just how you measured the drug, the methods. So this is very cut and dry. You start off with 2.6.4 with a description of the non-validated and validated methods that you've used to measure the drug. Now we're getting into the more science-driven, or less cut and dry kind of sections. First is the absorption section. And this would have all the absorption data that you've collected in your non-clinical studies, animal PK bioavailability, permeability, perhaps some discussion of a human bioavailability experiment, if that's been done, perhaps some discussion of BCS classification as well in this section. Impact of efflux transporters, and then again the more detailed on additional animal PK studies, including ascending dose studies, repeat dose studies, any specialty studies with pH or food effects, and formulation studies. So this is kind of the normal type of data that would be included in the absorption section. Disposition section would include any kind of tissue distribution studies that have been done, including target organ information, distribution to target, tissue retention and accumulation information, transporter-mediated distribution. Other studies like plasma protein binding or binding to albumin or alpha-acetyglycoprotein, impact of renal or hepatic insufficiency, and then any potential displacement interactions. Other studies in this section would be blood to plasma ratio and placental transfer. Moving on to the metabolism section. This is a section that kind of grows over time during the characterization phase. And a key part of this is the characterization of human metabolites. A big part of this section is the complete understanding of circulating metabolites in humans. This is kind of in line with a safety testing of drug metabolites guidance, the MIST guidance that kind of prescribes the level of understanding that you need for circulating metabolites in human, how you have to characterize them in non-clinical species, as well as in vitro tests. So the profiling of human plasma, determination of key metabolites, there's definitions of what a major metabolite is, and kind of the studies that need to be done to characterize that major metabolite, qualify it in toxicology species, as well as characterize it for any potential off-targets and on-target effects. Finally, data on enzymes responsible for metabolic clearance, including the fractional contribution of key enzymes, are an important part of this section. Excretion, the excretion from a mass balance study, and this is another key part of the development section. The full characterization of the drug molecule is the administration of some sort of labeled version of the drug, so it can be traced completely, including the excretion of it. So excretion from this mass balance or ADME study, parent versus metabolite, urine, feces. Other things would be transporter-mediated excretion, whether there's any kind of facilitated urinary excretion, anything you might know about facilitated biliary excretion, and finally secretion in the milk, be typical in the excretion section. Drug-drug interaction section. This will be a key part of the second talk today. And I won't go into this too much in detail, but the key parts are really characterizing the NCE as a victim or a perpetrator with DDIs coming through enzymes, CYPs or UGTs or other enzymes, or transporters. So characterization of both the perpetrator and victim potential for the drug, for the compound, and then how that actually, how that information matches with what has been found out in any kind of clinical DDI studies. Information on key co-medications, again with cross-references to clinical studies. And all this is covered in another very key guidance on in vitro drug-drug interaction studies. So that's kind of the modules, the key modules for where you want to be at the time of filing for a new drug. So let's move on and talk a little bit about what would be included in typical IND filing packages. And it is the same information that's necessary, but obviously in the IND stage, much of it is predictive of what's going to happen in humans, rather than actual characterization of what's going on in humans. So it is a little bit different in flavor, and some of the information in the IND is actually not going to be that useful after you start getting human data. So it has a little bit of a different flavor, and I'll go into some of those aspects now. So when you start to get ready to generate the information for an IND ADME package, what are the considerations and the drivers for doing studies? Some of this is really to support toxicology studies, and some of the studies that we'll talk about are really important for species selection and interpretation of toxicology multiples. So that's part of the information. The biggest part really, though, is to really try to understand and get a handle on what are the risks for drug attrition, for drug failures due to unacceptable ADME properties. So this is very important, obviously for the sponsor. At some level they want to know this. But for small companies, it's also very important for partners if they're talking to partners about perhaps co-development or actually sale of the asset to a partner. They'll be very interested in these risks of drug attrition. Now, large pharma companies usually will do all of the studies that I will kind of talk about in the next couple of slides, because they're very interested in trying to completely quantify the risk of drug attrition. But smaller companies, I think it's a much different kind of calculation to figure out what exactly needs to be done. So some of these risks are the potential for DDIs, polymorphic metabolism. What's the human dose projection look like? And does that projected dose meet expected target product characteristics? Are you able to actually deliver that drug in a way that will be fitting for that target profile, which for a lot of drugs is a once-a-day oral drug? Does that projection actually meet those criteria? Is the drug expected to have a low therapeutic index, and is that kind of drug going to be a potential success in the marketplace? Is there a lot of competition there or not? Another reason that you might want to think about this is, and this is even more common nowadays, that companies are trying to get into patients much earlier in human trials. So perhaps right into patients, or perhaps some normal healthies, and then a quick transition into patients. And if you're going to be into patient trials, you'll want to have much more information on potential liabilities early, so you can safely dose those patients. So some additional questions to address, and some of these I've already talked about, this question of normal healthies versus patients. In normal healthies, it's quite easy to just not allow any kind of other co-medications into the trial, so you can control what's happening. With patients, it's much more difficult to do that, so that's a key question, and how early you're going to include those. Are there any co-medications in those early trials that will almost certainly be necessary for people in the trial to be on at the same time as your medication? Is there information on necessary co-medications? These other characteristics that might dictate how much you need to characterize a drug, or what kind of indication you're going for. Is it really high unmet medical need? And is it an area it's kind of unique and probably you'd be willing to accept a lot of risks going into that a drug with liabilities could probably still be successful, so there may not be as much of a driver to do those kinds of characterization studies. What's the competitive landscape look like? Are there already drugs on the markets or in late development that have a certain profile that you might have to profile against and have a superior profile to? All those kinds of things kind of dictate how much information you might want to generate up front. So I've kind of broken things out to try to move through different studies into different categories here. And some of these will just kind of help frame this discussion of what kind of is probably a required component of 2.6.4, and where do you get more into the gray zone of what is probably at the sponsor's discretion. So these Category 1 studies are the studies that talked about that support the toxicology study. They are very important to really understand the drug and the safety multiples and things like that. In most cases, they'll be part of the filing. There are a number of other studies that have kind of lumped into this Category 1 that are descriptive studies about the distribution of the drug, the excretion of the drug in animals that perhaps are not as completely necessary. Again, Category 2 studies, kind of the same thing. These are studies that have categorized as having reasonable translation from non-clinical to human, and they predict kind of high impact liabilities. So this would be a subset of the drug-drug interaction type studies, and include things like CYP3A inhibition induction, OATP inhibition, things that have big drug-drug interactions that could be very important, and could be liabilities that would be difficult to get over with a drug. There's others in this kind of category that have reasonable translation, but they predict lower impact liabilities. I've just listed here the inhibition of perhaps some transporters, and just called out here inhibition of OAT3, where you could have a drug-drug interaction, but it's probably not going to be a very large, a large impact interaction, and may not cause such a problem. But finally, this last category is high impact liabilities, where there's a lower fidelity of translation. And these are some of the liability screens that are often run in discovery, and are difficult to actually make decisions on. And a lot of these are around predicting different kinds of toxicologies, and specifically drug-induced liver injury. And these are a couple of the screens that are often done to predict drug-induced liver injury, but are kind of difficult to really translate into actions in the clinic. So this has really been covered, I think, sufficiently already. These are these Category 1 screens, and they're important for species selection, and they revolve around different ways of understanding the toxicology, either with the protein binding. Obviously, toxicology studies have to be supported with exposure in those species, so pharmacokinetics are important to make sure that you're getting exposure. You also want to ensure that you get metabolites that, at least at this stage, what you know about metabolites from in vitro studies are reasonably covered in, or you anticipate they'll be reasonably covered in animal species. And then this interspecies protein binding. These characterization studies, the tissue distribution for drug distribution to target tissue, perhaps to aid in PK and PD understanding. This is the kind of study that's often done very extensively for CNS targets, for tumor targets, where you have a specific tissue that you're very, very interested in understanding distribution to really understand your efficacy better. But there's also a chance for drug distribution to off-target tissues, and this is to aid in liability assessment. Sometimes this could be an organ like the liver, and high distribution to liver may lead to the potential for liability there. And then finally, these excretion studies. The liability assessment around drug-drug interactions. Again, the DDI potential perpetrator and victim for the high-impact transporters. This, again, is covered in this drug-drug interaction studies guidance. This will be the topic, again, of Vlade's talk in a couple of minutes. I will just add that these studies are standard offerings at many CROs, but they really are often not exactly standard studies, and they do have to be carefully run and carefully interpreted. So I think it is, especially if you get to the IND stage, it's a time where hopefully somebody that has a reasonable background in these types of studies is helping you in the design and interpretation of them, because they're not exactly as simple as they may be portrayed by looking through a catalog at a particular CRO. Again, the PK relationships, polymorphic enzymes, understanding of how that might impact metabolism. Another part, so we'll get into this a little bit more in the end, the PK prediction from animal pharmacokinetics and different in vitro sources to come up with a prediction of human PK. Marrying that with the target plasma profile to come up with a projected human dose and how you might reach that with both a dose level and a frequency of dosing. This is not, this kind of assessment is not completely necessary, but it really adds considerable weight to the overall IND package. It's kind of a part of most, I think, modern IND packages where you have some type of human PK prediction and human dose projection that really helps both the design of the clinical protocol and interpretation of toxicology studies. These are I guess the lower impact enzymes, and I've just included a note here. If there's a need to perhaps conserve resources and budgets, these are the kinds of studies that I think some sponsors might choose to defer to post-IND activities, where after you have some level of understanding of human pharmacokinetics. Finally, the liability assessment and assessment of drug-induced liver injury. This is a little bit about how these modules kind of interact here. The 2.6.4 non-clinical PK, interactions with the pharmacology section and the non-clinical toxicology section. A lot of these have already been gone over. These are the kinds of things that would be at least cross-referenced, or there would be inclusion in one section or the other. One section here, like safety pharmacology within pharmacology, it's often good to have some sort of human PK projection to understand what safety pharmacology findings really mean and what they might mean when you get into those human trials. Finally, the interaction with the clinical protocol and how you're designing that first human study with a dose escalation scheme. So section 2.6.4 is really key to not just understanding ADME, but also understanding these other parts of the IND document. So I'll talk a little bit more. I think timing is still going well. And sorry, I should have said that we could entertain questions anytime during the talk, but I really haven't given much of a pause to do that. We'll have some time for questions afterwards. The human dose projection. So the science around human PK prediction from different sources has really, I think, blossomed over the years. And there's multiple ways of doing this. And there's very reasonable literature also on how to do this. Multiple methods listed here, just from in silico all the way through in vitro, in vivo correlations, the scaling from animal PK, finally PBPK. Sort of, I think the state of the art is to use multiple different types of approaches here, and then just see how they correlate with each other, how they coalesce. Hopefully they do. Correlate well, and you can come up with kind of a global prediction for human pharmacokinetics based on a lot of these different tools. The animal PK and in vitro and vivo correlations are the most common ways that this is done. And like I said, there's very good literature that helps in the conduct of these kinds of scaling exercises. Second part of this, step 2, is prediction of the human efficacious plasma concentrations. So along the way, and you're generating your efficacy data. Hopefully, there's been some understanding gained during that process of pharmacokinetic, pharmacodynamic relationships. So you'll have some sense of what the pharmacokinetics are in the animal species, the efficacy species that are driving the pharmacodynamic and the efficacy outcomes. So at the end, you can have some sort of PK profile that best describes that observed pharmacodynamics. There's a target PK, and the target that you ultimately want to reach in a human trial to see efficacy. Different kinds of measures of this often are just a straight AUC measure, perhaps a time over an IC50, trough coverage of an IC50. These are just typical kinds of pharmacokinetics that might lead to the best efficacy prediction, and you can then be translated into your target profile. Finally, you take the projected pharmacokinetics, marry those with the projected the targets, and come up with the dose that actually gets you to those targets. So that's kind of the dose projection exercise. This is just a manuscript from a number of years ago that kind of takes us through this process. This was a hypertension drug, and they had hypertension models, so used preclinical data. There was actually a clinical comparator that was used in this study. So they were actually very, very nicely able to come up with the predictors for concentrations that would give efficacy, these EC50s. And then finally, they took those targets, used the allometric scaling pharmacokinetics to come up with a dose response, predicted dose response for hypertension. So that's the process in a nutshell. Like I said, there's literature on the dose projection as well as the pharmacokinetic prediction aspect. So final topic here I'll talk about is just the methods used to inform DILI risk. This is a very broad and complex topic. I'm going to try to summarize it a little bit in just a couple of slides. I did talk already about some of the screens that are often used as part of this exercise. But it is a very complex and kind of a multifactorial kind of area that there's a lot of gaps in overall understanding. So the idea of trying to do this is it's challenging, and it is something that I think you really have to understand what you're getting into before you start into the overall assessment, because I think it's kind of progressed to a place where you need to be able to describe multiple different characteristics of a compound to do a proper assessment. It's not easy to do with just kind of a single random assessment. So this is just the kind of the basic understanding of drug-induced liver injury or hypersensitivity risk. Like I said, it's it comes from multiple factors, and this is a depiction of the complete response that ultimately is producing DILI. If you can see all the different aspects of the slide, you have the engagement of the innate immune response, which is a kind of a direct downstream impact of cellular damage. And then there's the adaptive immune response too. And the best understanding of DILI is that it's coming from the intersection of both of the adaptive and innate immune responses that are kicking in to trigger this adverse event. So the screens that are really conducted are around understanding how the propensity for this innate immune response activation, the cellular damage. And many of these come from the generation of reactive metabolites. And they kind of come through different pathways of that. Some is direct protein, some of it could be oxidative stress, but they're a downstream impact of reactive metabolite formation. So that's one of the screens that is often done, is to try to characterize the reactive metabolite formation. Another is through kind of a cholestatic process where you have interaction with bile acid homeostasis, and that is another potential avenue. So a lot of the predictions are through screens that look at the BSEP inhibition. That's a bile acid transporter in the liver, and that is often conducted in a BSEP inhibition assay. There are multiple assays that can be done in conjunction with this that are also involved with bile acid trafficking. Reactive metabolite generation, like I said, glutathione trapping to look for reactive metabolites, acylglucuronide stability, kind of typical studies. But really to get a final determination is through a combination of the above results and many different results, kind of putting them together and synthesizing an overall risk. And this is a publication from AstraZeneca that tries to do this exact thing where you're putting together multiple different outputs from screens, and here are many of these screens that I've already covered, different cytotoxicity screens or reactive metabolite screens, and trying to give some sort of liability risk assessment on the chances for liver injury. This is, you can see here you have to generate a lot of data to do this effectively, and it is just a very difficult thing. That's why I've kind of said that this is the type of assay that's I think you have to think about before you really want to start into it, because it is such a challenging area.